Dando continuidade à segunda sessão, teremos agora a palestra do James, do Dr. James, que possui graduação em Biologia pela Universidade Johns Hopkins, mestrado em Melhoramento de Plantas e Genética Vegetal pela Universidade de Wisconsin e doutorado em Crop Science pela North Carolina State University. James é professor na North Carolina State University e desde 1999 também é pesquisador genético no USDA, trabalhando com genética e melhoramento de milho. Agora passo a palavra para o James, que irá nos honrar com a palestra intitulada Resistência de doenças em milho, muitos genes e tão pouco tempo. As questões podem ser enviadas pela seção QIA de perguntas e respostas e estaremos atentos às questões para repassar ao palestrante ao final da apresentação. Dr. Holland, you may start your screen share. Thank you so much for agreeing to participate in this virtual symposium.
Okay, thank you. Are you seeing my screen now?
Yes.
Yeah, okay, cool. All right, thanks for the invitation. Very nice chance to talk to you about maize disease resistance. And you can listen to the first slide and you'll get the entire story, that there's a lot of genes involved.
Dr.
Yes.
James?
Yes.
Can you share your screen with
us?
I
thought I was. Okay. Let me try again. Oh, there we go. Sorry. How's that?
Okay, thanks.
Okay, right. So my title involves lots of genes. That's the story. Okay. Next. Okay, so I want to spend a little bit of time just at the beginning distinguishing between qualitative versus quantitative forms of disease resistance. Mainly because most of what people commonly think about, if they're not disease specialists, when they think of disease resistance, usually it's the qualitative form. This is the kind of form you basically have discrete classes. You have some plants that get no disease at all, and you have other plants that can become highly infected. So this is the difference between immunity versus susceptibility. And the immunity is conditioned by the hypersensitive response. And I'll talk a little bit more about that in a second. And we'll get a little bit into the molecular biology, not very deep, but this is caused by what's called effector-triggered immunity. And it's the key thing here is that this response of the plant is specific to proteins produced by certain pathogen isolates. So it's very finely tuned, only specific pathogens and only specific isolates of that pathogen will be recognized and trigger this hypersensitive response. In contrast to that is quantitative disease resistance, and that's where you don't have any immunity typically. So in this picture, this looks like an immune response. Most of the things that I've worked on, you never actually see, as long as you get the inoculations working well, and it's not an escape, you will never see something that never gets disease. But you do have this continuous variation. So you have some things that are extremely susceptible and some things that are pretty resistant, although not completely resistant. Some of this, will, again, going into the molecular biology, this can be conditioned by what's called PAMP-triggered immunity. These PAMPs, these are pathogenic related compounds, but they're very generic. So lots of different kinds of pathogens will produce these things, and this response can get activated. So in terms of the genetics, the kinds of the distributions you'll see segregating in populations are very simple in the case of qualitative resistance. That's why it's qualitative. You have basically two classes. Phenotypically, the genetics are simple. You can generally map these genes, and many of them have been cloned. In fact, we know a huge amount about the molecular biology of these things because the genetics are simple, the genes have been cloned, and the pathways that underlie these forms of resistance are very well understood. And for the breeders, that means simple breeding procedures are also very effective. So you can back-cross these things. Marker-assisted selection is really effective. So the breeding's easy. The problem is they tend to be less durable. So they put a lot of selection pressure on the pathogen populations that cause those pathogen populations to no longer produce the effectors that trigger this immunity. And that's how the pathogens can evolve avoidance of these resistance mechanisms. In contrast, again, here's the quantitative resistance, and this is hypothetical, but you see something like a normal distribution. The genetics are complex. There's a lot of genes involved. So the breeding procedures are the kinds of breeding procedures you would use for things like yield or any other complex trait. Back crossing is usually less effective, and marker-assisted selection is not effective generally because there aren't any large effect QTL to select on. The good news is this kind of resistance tends to be durable. Again, it's aimed at sort of generic features of pathogens and not at specific individual proteins that pathogen can mutate quickly. Here's a picture from my colleague Peter Ballant-Curdy here, looking at the hypersensitive response in maize. This hypersensitive response, this is the thing that underlies the qualitative resistance, and it's activated by these R genes. As I said, many of these have been cloned. They typically are these nucleotide binding site, leucine-rich repeat type genes. And many plant genomes have hundreds and hundreds of these genes. And this is, it's a little bit interesting because this is the way resistance work is the cell where that HR is triggered kills itself. So it's a kind of suicide that's restricted to a cell. So the plant has this mechanism whereby when it detects the infection, the individual cell and maybe a few cells around that kill themselves, but that hypersensitive response has to be very tightly controlled. You don't want it to spread beyond that. The plant doesn't want it to spread beyond that. And obviously you don't want it being activated in the absence of the specific pathogen. This picture is actually a mutant in maize. This is the RP1D21 allele. RP1 is a rust resistance gene, major gene. It's isolate specific, but this D21 allele is an autoactive. It just goes off without the pathogen around. And so my colleague Peter has used this gene to trigger this immune response without the pathogen. And it's allowed him to study the interactions between these major resistance gene, this HR thing, and everything downstream in the pathroom. After that thing has been activated, somehow this line here suppresses the spread of that HR response versus this line down here. So this has the same allele, the genetic backgrounds are different. So you can see that even in the case where you have major single HR type resistance, there is some quantitative aspect to those as well that sort of modulates how strong the effect is. This is a pretty interesting area. I'd recommend reading this paper, and it brings up some questions that we don't really know the answers to, and that is, in this hypersensitive response type resistance is very common in self-pollinating species. So when wheat breeders heavily depend on this, rice breeders use this thing a lot. In maize, outside of mainly the rust resistances, there aren't any such useful resistances. Most of the resistance in maize is quantitative, and it's not this form. And so the question is, why is there this difference between maize and other species? And is that related to the outcrossing habit of maize? So anyway, those are just some things to think about. I won't dwell any further on it. A little more molecular biology, just very quickly, really not much detail, but I just want to bring home this point that hypersensitive response, that single gene type resistance, occurs when here's a fungus, but whatever microbe actually injects into the plant cell these things called effectors. So these are some proteins that are produced by the pathogen. And they are involved, these are things that the pathogen puts into the cell to help the pathogen overcome the defenses of the cell. But plants have these R genes that in some cases will identify these effectors. And once that binding has happened, that triggers this hypersensitive response. So this is the single gene type resistance. But as I said, even in quantitative disease resistance, we think this is part of what's going on. In some cases, there's maybe attenuated hypersensitive type responses. In quantitative disease resistance, you have something else going on in addition to that, and that is these responses to more generic compounds that are produced by lots of microbes, these so-called PAMPs, and there seem to be receptors like in cell walls and things like that that identify these things, and they set off different kinds of pathways. The phenylpropanoid pathway is one way. There's things like jasmonic acid, salicylic acid pathways. Those all get activated. There's also sort of passive defenses. There are, compounds that plants make that just are preventing infection in some cases. And you can think at even a higher level that it's not just within the cell, but it's also the structure of cell walls, cuticles, trichomes. Microbes enter through stomates, so actually stomate structures might be involved. There's basically everything going on at this very kind of high morphological level. These could also be aspects of quantitative disease resistance, and they wouldn't necessarily be simple, nor would they necessarily be active responses. They might just be that's how the plant is. Even things like leaf angle and leaf shape will affect how spores can land on the leaves and germinate and penetrate. And then what I'm going to talk about for later in the talk is ear diseases in maize, where there are fungi that get into the ear and cause disease on the ear. And obviously, some part of the resistance to that are things like husk covers and ear morphology. So they're not like these hypersensitive response things. They're a little more generic. So if you want to study genetics of quantitative disease resistance, the genetics are complex because there are a lot of genes. Their effects tend to be small. So it's difficult to map and clone these things. So one approach we took was to just try to do very large scale genetic mapping. And this was to use the Maize nested association mapping population. If you haven't seen this before, it's just a population structure whereby we have 25 diverse inbred lines representing all kinds of different maize types, many tropical varieties, some northern varieties, sweet corns, popcorn, all these sorts of things get crossed to a common reference line. And from those 25 F1s, we generate 200 recombinant inbred lines from each of these crosses. So the total population size is about 5,000. That gives you really some pretty good power to dissect even relatively complex traits. And so we applied this to southern leaf blight. This is, I don't know how you say it in Portuguese. It's Cochleabolus is the genus, Heterostrophus is the species. That's the Latin name. And again, there's no complete immunity. You get some things that look like there's very little disease on them. There's probably a little bit, but it ranges. And we tend to score these things on a kind of a breeder scale, 1 to 9 type scales. So we phenotype the NAM population in multiple environments. The population was genotyped, so we could map QTLs. And then also the founders had been genotyped more densely. That allowed us to do projection of the founder SNP variants onto the populations and do a genome-wide association study as well. And just very quickly, this is one kind of statistic we'll use for reporting statistical support for a QTL, this RMIP thing. I won't get into the details, but the higher up you are, the more evidence there is that the variant that we're picking up is associated with the trait. So these arrows and these names here, these are all variants that are either in or very close to genes with some known function. So we've written out the gene names here. There's a few of them that we don't know what they are, but a lot of them had known functions. And a lot of these things look like they could have some role in disease resistance. And you'll see actually some of them are in those LRRs. That's like a major resistance gene, but a lot of them are different. So, and Peter Ballant-Curdy and his group picked this caffeoyl-CoA O-methyltransferase. This gene's involved in the lignin pathway, so it's very different from those major hypersensitive response things, and decided to do functional validation. So this is a GWAS, it's an association, you can be wrong. It's not proof that this gene's involved. The proof required a lot more work. And basically one further piece of evidence is to do this kind of fine mapping study, and I won't go into a whole lot of detail, but basically get that QTL allele into a common background with a more susceptible type. So the only difference between these two lines is one introgression, and that introgression clearly increases the resistance. And then from that, you cross these two individuals and you generate a lot of progenies, identify recombinants, and if you get lucky and you have enough markers and you have enough recombinants, you can fine map the QTL from this sort of big region down into this spot here. And there's only two genes. One is this F box and the other is this CCOA MT2, which lined up, this thing lined up with our GWAS-association. There was some evidence that ruled this gene out. I won't go into that, but there's good evidence that this gene actually is involved in southern leaf blight resistance. And that evidence comes from what are called mu insertion mutants, and a mu is a transposon, and if you can get that transposon to jump into this gene, you can change the expression and you can change the effect on resistance. But also we have the gold standard. I didn't do this work. This was Chin Yang and Peter Ballant-Curdy that actually did all this functional validation, and they made transgenics. And you'll see that there's a series of these transgenic events here. And each of these has a paired transgenic positive versus non-transgenic from the same original event. And you'll see that in most cases, well, there's a significant increase in resistance. Some cases there's not, but if you look at the expression levels of the gene, the cases where there wasn't a difference, well, there really wasn't much expression of the transgene. So everywhere you had good expression of the transgene, you had a significant effect on southern leaf blight. So that was pretty good evidence. The point here, though, is that this was a tremendous amount of work. Okay, a huge amount of work went into identifying one gene that, if you look, it's changing the phenotype scale here is our 1 to 9 scale. It basically changes the score about a half on our level. If we go back here, it's that difference right there. So that looks pretty good. In other cases, it's a little more subtle. So it's on our original scale, it's sort of halfway between here and there. So, you know, it's real, but the effect is relatively small. And so as a breeder, so this is great from the biology point of view. We've learned some biology, and we've learned that these quantitative disease resistance genes can operate through lots of different pathways. But for the breeder's point of view, you know, this is essentially a fairly small effect gene. You know, it's a QTL. You could use marker-assisted selection and introgress it, but you're not going to get that much effect from that one gene. So it's a lot of work for maybe not that much impact in terms of breeding. So I'm going to switch gears here, and so now I'm going to talk about Fusarium ear rot. And as I mentioned, that the diseases that affect the leaves, these foliar diseases, are actually pretty nice to do genetics on, and we have fine mapped and identified some functional variants that underlie them. But ear rot doesn't work that way. Ear rot is very complicated. It's caused by Fusarium verticillioides. It's one of several Fusaria that will infect corn and maize, and cause damage to the ears. It causes this starburst. When it's not real bad, it causes the starburst phenotype. These little white streaks on the kernel, those are actually hyphae of the fungus growing underneath the pericarp of the seed. When you have the conditions just right, you can have things where the ears get completely destroyed by this fungus. It also produces stalk rot. It also has a seedling. It can kill seedlings. So it causes a lot of damage. And the real complicated thing here is that we don't know the biology of this thing very well. We don't know any resistance genes. There is no immunity. We don't know any genes. So the rest of my talk is going to be, how do we breed for resistance, given that we have all these very nice genetic and genomics tools available to us. But if the disease resistance is very complex and we can't identify the underlying genes, what are we going to do with that kind of information, and how do we use it for improving breeding? One other thing this fungus does, that's going to make the story even worse, is it produces a toxin called fumonisin that gets in the grain, and it will kill animals, and if humans eat enough of it, it appears to cause cancer. This is a study where they looked at grain samples, I guess that were on the market, and tested them for different mycotoxins, and you'll see fumonisin was the one that shows up most frequently across the world. And so it's pretty much everywhere maize is grown, you can get this fusarium ear rot, and you can get this fumonisin contamination. The other weird thing is, why does it produce this? We don't really know. It doesn't appear to be a virulence factor. Okay. It's also highly influenced by the environment, and this is a map of the USA, and you know, taken a little bit out of context. So I'm down here in the southeast. For example, here's Washington, D.C. New York is sort of up here. New York City is up here. In the Midwest, this is the corn growing region. Temperatures are cooler. You do get some of this fumonisin, but it's not as bad as when you get into the more southern and hotter regions. And the worst place for fumonisin in this experiment, which is the biggest one that I know of, was in eastern North Carolina, where we are. And this is well above what our government recommends for grain going into food or feed. So it's really a problem. Again, I won't belabor this, but the inheritance is complex. There's no major genes. And the other thing is, it's actually pretty labor intensive, the phenotype. So I spent a lot of time trying to do genetic analysis. We've done QTL mapping. We've done GWAS studies. So we've done biparental crosses. We've done, you know, diversity populations. And basically, you can identify some QTL. They tend to be different in every population. None of the effects are very big. You can identify things with GWAS. None of the effects are big. And they tend to be different in every sample that you do, probably every environment that you do. So again, it just shows that, you know, there are genes here. It's genetics, but you know, the genetics is pretty complicated. The other thing we know is that we know it's genetic because phenotypic selection works, but it takes a lot of effort. So you have to grow enough replications, you have to do enough environments. We will inoculate artificially to make sure we get good disease. Under those conditions, the heritability is generally pretty good. It's 50% or above. The big problem is the labor-intensive nature. It's just so much work. So even though we know that there's not specific genes we want to use marker-assisted selection for, could we use this genomic data that's available to help us do selection? And the obvious thing here is to try genomic selection. And this is just to illustrate the amount of labor that goes into doing this. And we're kind of on our own now. We don't have pathology colleagues to help us anymore, so we have to culture all the isolates. You have to drive them out to the field, and with these heavy backpacks in the summer, and inoculate into the silk channel, and we do a second, so 2 trips to every experiment station, 2 different kinds of inoculation. Have to harvest everything by hand, comes back to the seed lab and gets lined up. Every single ear has to get scored by eye. Now, maybe you can have some phenomics thing that will automate this. That would be great. Right now, we don't have it. And then to do fumonisin, it's yet more work. You have to grind those. You have to get the grain off, shell them, grind up, and then run ELISA assays, which are now like $3 a sample. So it's getting expensive. as well as it takes a long time. We won't get all the information back usually until the spring or early winter after the year we've grown them. But phenotypic selection does work. We developed a recurrent selection mapping population, a recurrent selection population. And that was segregating for, different quantitative levels of resistance, and also hopefully was segregating for decent agronomic material. And we did 3 cycles of phenotypic recurrent selection among S1 lines. And here's ear rot, here's fumonism. The cycle 3 distribution is the blue. The original population's here. So you can see that we definitely pushed ear rot lower. We pushed the fumonism levels lower. We did not ruin yield potential. So it worked. And the other problem is each cycle of selection took 2 years. So that was 6 years of breeding just to move the ear rot, 13%, something like that. So then we thought, okay, we can use this same population though, this cycle 3, where we ended the phenotypic selection, and evaluate a large set of S1s from that population, both genotypically and phenotypically, to develop a genomic selection model. So the first thing we wanted to do is to figure out, is this actually going to work? And the way we did that is we actually took that whole set, that whole cycle 3 population, and split it into 2 parts. We had a training set that was evaluated in 2 years, and then a test set, which was different lines, evaluated in a different year from the training set. This was work of Tiago Marino, who was a PhD student here. And the lines were genotyped, and then he built genomic prediction models of various flavors and evaluated them in the training set. And just to show you, I'm not going to go through any details on the markers. We did a lot of quality control of marker data, a lot of quality control. So we didn't start with 6,000. We started with much, much more than that. But we got down to 6,000 that were pretty good, nicely behaved markers. And when you look at the population structure here, you can see there's a little bit of relationships going on, when you sort these things by similarity. But basically, it's a random mating population, so there's no large-scale population structure. We looked at LD. I don't show you any details here, but the LD is extensive enough where genomic selection should work for some time. And then what we did was here's an example where we used a GWAS model based on one set of lines in the first 2 years, and we have their predicted values on the x-axis. Sorry, yeah, so the training model is built on 2014-15 data, but this is the prediction for the lines we tested in 2016, which is a different set of lines. And there are 2 different colors. Again, I don't want to get into those details. They're just sort of 2 different ways we had of doing it. And you can basically see, yeah, there's some correlation for fumonism. That's pretty good. Maybe not quite so good for fusarium. But basically, there's sort of reasonable prediction accuracy on these things. So we'll just talk about the blue bars. That's prediction accuracy. So that's scaled to the square root of the heritability. And then we tried all these different models. Again, the details aren't that important. The relatively simple GBLUT models work just fine. The Bayesian stuff in general doesn't really help. And we tried some really fancy machine learning models, and they were terrible. So this is an indication, again, that the genetics is just very complex. You have a lot of genes involved with small effects. And I'm almost done. So just a couple of points I want to make here, just to keep in mind. Genomic prediction is not as good as phenotypic selection if that phenotypic selection is based on lots and lots of field data. Okay, so we're not saying we give up doing phenotyping. We need that phenotyping to build the models. The advantage of genomic prediction is that we can do it faster than phenotypic selection. So over time, we hopefully will get greater response if this works. So the idea here is that the phenotypic selection, the way we were doing it with recurrent S1 selection, took us 2 years per cycle. With genomic selection, in theory, you can implement that. We can do 2 seasons a year probably companies could do 3 seasons a year, so you could get many more cycles of genomic selection. So this is what we've done. We then took the data from both the training and the test sets were in that original base population. So our training model is really about 500 lines now. Put that together, and then we basically bifurcated the population. We selected the best parents for going based on the phenotype data only, and that went into the phenotypic selection. And we just went through that again for another cycle. Or basically 2 cycles. So it took us several years here, and we had to run another round of phenotyping in multiple environments. And where we are now in 2020 is we have a random sample of the cycle 5 phenotypic selection lines in the field now. Meanwhile, at the same time, we ran a parallel genomic selection project where we used the training data from back here, and we just selected on single plants, because that's the power of genomic selection. The details get a little complicated here because managing these things in a nursery and trying to get the data back on time is non-trivial. If you're at a public institution where it's just you and yourself and maybe a grad student doing it, we don't have huge teams. So we're not super efficient at this, but we managed to get 1, 2, 3, 4, 5, at least 5 sort of bursts of genotypic selection at the same time. We got 2 rounds over here. So this is in the field this summer. I have a new student, Eric Butoto, who's evaluating this. And I'm about out of time, so I'm gonna just mention, I don't have the phenotypic data yet, but it's in the field right now. We do have marker data from the genomic selection, and these purple lines were the original parents of the population. The red dots are that cycle 3 population we started from, and you can see as soon as we started genomic selection, we pushed the allele frequencies very quickly and very strongly, and that's a little dangerous. So we know we've done something. We've had some effect here. We hope it's in the right direction in terms of the phenotype. The other thing is, now you can see we're in this cycle 6 out here, and if you're trying to continue to do genomic selection sort of out here in the genomic space where the training was built over here, we're probably not going to be effective any further. So we're basically stopping. We're going to evaluate this summer phenotypic versus genotypic selection. If the genotypic selection worked well, then we're going to have to do more experiments and recalibrate the genomic prediction model to keep moving forward. So that's where we are now. Again, it's in the field. I can tell you next year if this thing worked or not. And then this is just a little bit of theory. I won't go into it. There's a nice paper from Melkinger's group on how fast the prediction accuracy drops in these kind of recurrent selection systems. And it looks like in general, you can still get some gain from selection for 4 or 5 generations after the initial model build. After that, you probably need to retrain. So I want to thank all the people who did all the hard work. I want to thank Peter Ballant-Curdy, who's my USDA colleague here, who's done a lot on the the genetics and the functional validation. Rebecca Nelson and Randy Weiser are other colleagues that we've worked on for quantitative resistance. And then Jason's my technician. Eric, Tiago, David, Charlie, Megan, Leilani, those were all grad students who developed these populations and phenotyped them and did all the genetics. And I will stop there. And I guess I'll take questions.
Okay. Thank you so much for your presentation, Dr. James. Now we have a moment for a question for the public. Dr. Holland, you said that most of disease resistance in maize are quantitative. However, it is possible techniques such as transgenics and gene editing to tackle and sort of transform the quantitative disease resistance into qualitative disease resistance.
I wish. You know, who knows? It would be wonderful if that were true. I don't think that's going to happen anytime soon. Again, because the forms of resistance that are quantitative are complex and involve many components. There is no one single component that will give you immunity in the quantitative type disease resistance. So, and to my knowledge, there's no example in maize. I can't even think of a major crop where there is a transgenic disease resistance. You can think of that there are virus resistance transgenics that have worked, but basically transgenic disease resistance has been a real big failure. So I don't see that changing, and I don't know how gene editing is going to solve that. Although it would help in these cases where if you could identify some of the larger effect QTL down to the gene level, and if you knew those variants, that obviously that might allow you to introduce resistance variants into into elite backgrounds more quickly.
Okay. We have more one. Dr. James, could you talk a little bit about your kind of population? Have you been using GWAS studies for qualitative resistance?
Yes, we have used, actually I think that question came in before I showed a slide on that. So I think I may have answered it after it was posed, but so we've done all kinds of things. We've done diversity samples where we have tried to evaluate every single inbred in the USDA seed bank, and that is 2,000 some lines. Some of them didn't flower and produce ears, so I think we got data on something like 1,700 of them. That's basically, we sampled everything we could possibly sample that was an inbred, and we did GWAS with that. We've done GWAS with the sort of core diversity panels, and we've done biparental QTL mapping in multiple populations. And then again, we've done the NAM analysis for fusarium in, I think it was 4 of the populations. And we have mapped QTL in that. We just could not, again, the labor is so intensive, we could not do the whole thing. But we basically identified the founders that seem to have the best resistance. And we worked on those 4 populations. So we've done all kinds of things. And no matter what we do, we never find a major resistance gene.
Okay.
That's the sad part.
Thanks. You said you are improving